Hello, welcome to the microscope lab. This is what you would have seen walking in had we been allowed to be here in person. And these are some of the materials: the photographic atlas, the fixed slides, including the bacterial slides, and the different shapes for different smears. This is the anabina, the long chains of filamentous algae. And then here are the materials we use to prepare our wet mount slides. The allodia, which is that really thin, oh, here's some on the tweezers already, but you can see how thin those leaves are, which is why it makes a good specimen. Here's an onion. This was an optional one. Here's the potato, which we made a real thin slice of and added the iodine. And here's a carrot. So here is an overview of the parts of a binocular compound light microscope that I wanted to show you about. So we're going to start at the top, and this is called a binocular, meaning two ocular lenses. You can see them here. They're separated by a little distance, and you can adjust the width of the spacing of those ocular lenses right here. And these ocular lenses in and of themselves have a magnification of 10. So anytime you're looking through those two ocular lenses, even if you didn't have any other lenses, you'd have a magnification of 10. They sit on a structure called a headpiece, and you can actually rotate this headpiece around a little bit, and I'll show you how that's done. If I can set the camera down. So you can maneuver this little screw right here, and you can rotate this headpiece around. And that's helpful when you're working in pairs and you want to show your lab partner something without moving the whole microscope. The next set of structures have to do with this next set of lenses. And so this is what makes our microscopes really kind of cool, is that you can use these 4 different objective lenses which rest on a nose piece, and you can rotate them around. And as you increase magnification, you shouldn't have to adjust the coarse focus. And that's because these microscopes are parfocal, meaning you can keep them on the same coarse focus while you rotate them around. So this shortest lens is the low power objective. This next one is the medium power, and this next one is high power. And you can see the numbers right here. This number 4 means that this objective lens right here, this low power objective, has a magnification of 4. Now to get the total magnification, you would need to multiply that 4 times the 10 that we have with the ocular lenses right here. So that total magnification, when the objective, the low power objective is facing down and clicked into place there, would be 40. So with this microscope, we have multiple different magnifications we can use. The stage is where we want to rest our slide. So we want to put the slide into this little clip right here. And I'll show you an example of that in just a second. And that allows us to maneuver the stage in its position relative to the lenses using these coarse and fine focus, and also using these stage adjustment knobs. So as you move the slide adjustment knobs, as you move them with this knob right here, you can move the slide in that clip forward and back and right and left. And that's really helpful when you're looking for something using your microscope. So let's use a blank slide here. Here we have a box of blank slides, and let's just clip this in. I'm going to put it on the stage and clip it in, and now you see the slide clipped into place, ready to use. Another couple of parts of the microscope that you should be familiar with are the condenser, which is underneath here, which you actually can get a pretty good view of. This condenses the light as it makes its way from the light source. And there's a couple of adjustments we can make to that. We can use the condenser knob over here on the other side of the stage to move the condenser closer or farther from the underneath part of the stage. And then we can also use this iris diaphragm lever to control the size of the opening. Both of those things allow you to play with the light that's coming through and increase the contrast sometimes of the specimen you're looking for. And also if you have something that has a little bit of dimension to it, like a thicker specimen, you can play with this position of this condenser, and that allows you to manipulate where the light is focused and what you see. And then we have the light source down at the bottom. That's controlled by this on-off switch right here, which is the first thing that you would turn on, and then the intensity dial over here. So when this is on, then you can manipulate the intensity of the light, and that's something you might need to increase as you increase magnification, because when you increase magnification, you decrease your field of vision and decrease the amount of light that's coming through. A couple other basic parts are the base, the bottom that the microscope is resting on, the arm, this is where you would carry the microscope. And I think that's the big ones. So, sorry you weren't able to do this in person. Maybe next time.